The experimenter themselves remained blind to the taste condition. THEY were asked to dilute some water with another substance which they could choose from a group including sugar, apple juice, lemon juice, salt, vinegar, or hot sauce. THSA options form a nice scale running from pleasant to unpleasant, and so constitute a measure of the participants' aggression toward their critic. THE insulted participants showed greater left frontal activity than the control group. Within the group of insulted participants, the strength of left hemisphere activation correlated positively with how angry they reported themselves to feel and with the unpleasantness of the drink they concocted. Evidence that anger is approach-motivated comes from other types of study too. Transcranial magnetic stimulation (TMS) is a way of changing the polarization of neurons in certain areas of the brain. Slow repetitive pulses can reduce brain activity, so when repetitive TMS is applied to the right hemisphere, the left hemisphere increases its activity. When this is done, participants show increased attention to pictures of angry faces. D'Alfonso, Van Honk, Hermans, Postma, and DeHaan, 2000. THR or other less intrusive ways to increase left brain activity. A simple way of producing hemispheric differences in activation is to make a phi street with one hand. Contracting your right hand increases left hemisphere activity. THO's participants whose left brain was made more active by squeezing their right hand gave longer and louder punitive sound blasts to another participant who had previously insulted them. Peterson, Shackman, and Harmon Jones, 2008. Beyond the laboratory, anger correlates positively with psychometric scales measuring a person's approach motivation and negatively with their avoidance motivation. Harmon Jones, 2003, Schmitz and Cuppins. 2005. At a physiological level, anger increases muscle tension, heart rate, blood pressure, muscular blood flow, and body temperature. THSA are the familiar components of sympathetic nervous system activity preparing us for exertion and combat. So, do men simply get angrier than women? THR or different data source we can consult, but they all converge on the same answer: no. Some information comes from the study of anger as a personality trait which is measured by paper and pencil multi-item tests. Respondents rate their agreement with statements that describe a general tendency to experience anger, such as, "Sometimes people bother me just by being around." Archer 2004 brought together 46 of these studies in a meta-analysis and the results were clear. THR was no sex difference at all. See also Kring, 2000. Perhaps men experience anger more frequently. Some studies find that there are no sex differences in frequency. Brebner, 2003, Simon and Nath, 2004, with both sexes experiencing anger about 6 times a week. Frost and Averill, 1982. Others find that women experience anger more often than men. Mierowski and Ross, 1995, Ross and Van Willegen. Fear 85. 1996. Perhaps then, men's anger is more intense. Experimental studies, which elicit anger through the presentation of Phi-LM clips or slides, do not generally find sex differences in reported anger intensity. Kring and Gordon, 1998, Wagner, Buck, and Winterbotham, 1993. Experiments that elicit descriptions of hypothetical or remembered emotional reactions to provocation also find few overall sex differences in anger intensity. Manstead and Fisher, 1995, Milovchevich, Howells, Drew, and Day, 2001, and, where they do appear, they favor women. Brody, Lovas, and Hay, 1995, Fisher and Evers, 2011, Harris, 1994. International surveys also find no sex difference. Brebner, 2003, Fisher, Mosquera, Van Vianen, and Manstead, 2004. Where a sex difference is found, it is women's anger that is more intense. Brebner, 2003. Australian sample, Simon and Nath, 2004. So, if anger is not the source of sex differences in aggression, could it be that the sexes differ in their braking mechanism? Fear. Fear. Like anger, fear is a fundamental emotion that we share with other species. Joseph Ledoux, 1998, page 128, who has spent his life studying the fear response, encapsulates its function perfectly. Fear is designed to detect danger and produce responses that maximize the probability of surviving. His studies on the architecture of the fear circuitry reveal the critical role of speed in getting us safely away from threats. When danger presents itself, the information is dual-tracked to key brain areas. Fast route takes the crude sensory information that has arrived at the thalamus from the eyes and ears, and delivers it within 50 milliseconds to the amygdala, a small almond-shaped structure buried deep in the temporal lobe. Once activated, the amygdala's connections to a variety of other brain structures cause increases in heart rate, blood pressure, respiration, vigilance, cortisol secretion, and cortical arousal. Behaviorally, it triggers freezing and withdrawal in order to stop forward motion and pull the organism away from immediate danger. While this is happening, a second, slower, system delivers more detailed information to the amygdala from the sensory cortex. THs allows a more finely discriminated recognition of just what this threat is. The amygdala communicates with the frontal lobe that sends back commands for behavioral and physiological adjustments that may correct the initial, automatic, response. The whole process is a marvel of evolutionary engineering. Imagine this. You are sitting watching a late-night film on television, absorbed in the plot. Someone tries the front door handle. Even as you hear it, you freeze, your heart speeds up, your breathing quietens, your senses are on massive alert and highly sensitized. A split second later, your frontal lobes integrate more information. 86. High stakes and low risks, women and aggression. Your hippocampus sends memory information that your husband was out and should be returning about now. Your senses catch the sound of a key, burglars don't have keys. Panic over. You can breathe again and your heart rate returns to normal. Is kind of apparent overreaction may be physiologically wasteful, but the fear system works on the fiery alarm principle. Better too many false positives threats that turn out to be harmless than a single false negative and undetected threat that kills you. I have highlighted the fact that, for good evolutionary reasons, fear is an avoidance motivation. The evidence on brain lateralization confirms this. People who normally have higher right hemisphere activation report stronger negative responses to fear-inducing FILMS, as well as other avoidance-motivated emotions such as disgust. See Harmon Jones, Gable, and Peterson, 2010. Using TMS to increase right frontal activity causes participants to avoid pictures of angry faces which they had found engaging when their left hemisphere was activated. D'Alfonso, Van Honk, Hermans, Postma, and DeHaan, 2000. Individuals who are high in avoidance motivation show a stronger EEG response in the right hemisphere when viewing negative stimuli. Peterson, Gable, and Harmon Jones, 2008. But is there evidence that the sexes differ in the frequency or intensity of fear? If women's reproductive success depends on their own survival, we would expect that they would have highly sensitized fear reactions. And it appears that they do. In infancy, girls express fear earlier than boys. Nagy et al., 2001. They show more hesitation in approaching novel objects and greater distress. Gartstein and Rothbard, 2003, Martin, Wisenbaker, Baker, and Huhtinen, 1997. Rothbard, 1988. Around the world, parents report that boys show stronger approach behavior than girls. Carey and McDevitt, 1978, Xu, Sung, Stigler, Hong, and Liang, 1981, Maziad, Baudreau, Vierge, Capera, and Cote, 1984. Sex differences increase through childhood. Fredrickson, Annas, and Fisher. 1996. A Canadian study plotted developmental changes in personality, identifying one group of children that showed a profile of high and increasing fearfulness. The group was dominated by girls. Cote, Tremblay, Nagin, Zocolio, and Vitero, 2002. Among adults too, women experience fear more intensely than men. Brody and Hall, 1993, Crawford, Kipix, Onyx, Galt, and Benton, 1992, Fisher, 1993, Gallone, 2000, McLean and Anderson, 2009. International surveys have found significant sex differences in the intensity and duration of fear. Brebner, 2003. Fisher and Manstead, 2000. Women express their fear more intensely than men, both verbally and non-verbally. See Madden, Feldman Barrett, and Pietromonaco, 2000. While women are superior to men in accurately identifying all emotions, they show the greatest accuracy of all for decoding fear. Hall, Carter. Fear 87. And Horgan, 2000. One of the 5 key personality factors is anxiety or neuroticism. Women exceed men on this trait across the world. Costa, Terracciano, and McRae, 2001, Feingold, 1994. Fear has been taken into the laboratory in experiments that exploit the potentiated startle response. The paradigm goes like this. Participants watch slides that elicit different emotions, including fear. As they watch, a sudden blast of noise is delivered. Is causes an automatic startle reaction, a marked eye blink, which is more pronounced when the participant is viewing a negative, fear-inducing image. Much like in the cinema when a sudden gunshot or ghostly apparition is more frightening when the mood is tense than when it is more relaxed. The startle reaction can be detected by electrodes placed near the eyes, and other electrodes record negative facial expressions in the corrugator's muscle, the muscles between your eyes that contract when you frown. Women and girls show more corrugator's muscle activity than men when watching negative images and a stronger blink response to the noise blast delivered during the fear-inducing pictures. The also show greater sweating on the palm, indicating higher anxiety. Bradley, Cuthbert and Lang, 1999, McManus, Bradley, Berg, Cuthbert, and Lang, 2001. A phobia is a marked and persistent fear of specified objects or situations which the sufferer recognizes to be excessive or unreasonable. The Fear Survey Schedule, designed to measure this sensitivity to phobic objects, shows that females score higher than males. Arundel, Koch, Pickersgill, and Hageman, 1993. Gullone and King, 1993. To rule out the possibility that this was caused by factors such as gender role conformity or differential willingness to admit to fear, these were measured and their FECT statistically removed from the analysis. Arundel et al. 1993, Arundel, Mulkins, Koch, and Vollenbroek, 1999. Sex still remained a strong predictor, with women scoring higher than men on each of the F5A types of fear: social, agoraphobic, bodily injury, death, and illness, sexual and aggressive scenes, and harmless animals. The fact that sex differences in fear have a fundamental biological basis and are not simply the result of the way we socialize boys and girls has recently received further support from studies of testosterone. In a series of innovative experiments, testosterone was orally administered to women. Neither the woman nor the experimenter knew whether she had received testosterone or the inert placebo solution. Following this, the women were tested for alterations to their fear threshold. To examine whether testosterone would make women less attentive to threat, the experimenters used a task called the Emotional Stroop Test. Van Honk, Pepper, and Shutter, 2005. A series of faces is presented on a monitor, each with different facial expressions: neutral, happy, and fearful. The exposure time is so brief that participants are not consciously aware that they have. 88. High stakes and low risks, women and aggression. Seen anything at all. Each photo is colored to appear red, green, or blue. The participant's task is to name the color of the photo. Psychologists have established that the more strongly our attention is engaged by a picture, the longer it takes us to name the color. So, as expected, these researchers found that participants were faster in identifying the color of the neutral faces. When exposed to fearful faces but not happy ones, women who received testosterone were much faster at color naming, indicating that they were less emotionally engaged and threatened than the placebo group. To follow this up, another study used a version of the potentiated startle paradigm described earlier. Participants were periodically startled by white noise delivered through headphones and their eye blink response was measured. Hermans, Putman, Boss, Kopachar, and Van Honk. 2006. At the start of the experiment, electrodes were attached to their wrists and they were told that they would receive electric shocks. The would receive 3 electric shocks of increasing intensity, but they would never be shocked when the word, safe, was visible on the monitor screen. In fact, only 1 shock was actually delivered. Over the next trials, the experimenters measured their eye blink reaction to white noise blasts, both when, safe, was on the screen and when it said, danger. The expected that the, danger, signal would potentiate the startle response, and it did. Eye blink responses to noise blasts presented when, danger, was signaled were much stronger than when participants believed they were, safe. But when women had taken testosterone, their eye blink reaction was significantly reduced in the, danger, condition. In another study, testosterone administration was associated with heart rate acceleration to images of angry faces, a cardiac pattern associated with low levels of fear and a willingness to attack. Van Honk et al. 2001. Surveys, experiments, and physiological responses all suggest that women are higher in fear. And this may explain why men make riskier decisions than women. Hirsch, 1997. TH's sex difference in risky decision-making becomes larger when the risks involved are potentially life-threatening and when researchers study real-world risky behaviors, such as turning across oncoming traffic with barely time to make it, rather than cold hypothetical choices. Would you prefer a 50-50 chance of winning $100, or a 100% certainty of winning $45? THs is why psychologists have come to realize that fear responses may explain gender differences in risk-taking more adequately than the cognitive processes involved in the reflective evaluation of options. Burns, Miller, and Schaefer, 1999, p. 378. Traditional theories of risk assessment have overestimated the role of cognition and underestimated the impact of fear. Lauenstein, Weber, H.C., and Welch, 2001. While cold rational cognition can be useful for making impersonal and detached decisions, emotions are central to urgent decisions about approach or avoidance. Frieda, 1986. Unlike objective cognitive anger, fear, and aggression. 89. Risk appraisals, fear does not work on probability estimates such as likelihoods or odds. It works on an emotional all-or-nothing principle. Fear is sensitive to the possibility rather than the probability of negative consequences. Lauenstein et al. 2001, p. 276. If something just might be dangerous, avoid it. Fear favors cautious, risk-averse decisions. Lerner and Keltner, 2000, 2001, and, like that fiery alarm, it errs on the side of false positives. Anger, fear, and aggression. What happens when anger and fear are experienced simultaneously? Relevant studies are few and far between. Winstock 2007 studied them together in the context of hypothetical aggressive conflicts. He presented participants with vignettes in which they imagined they were approached by a male stranger in the street. The danger that he presented was varied in terms of the stranger's demeanor: easily scared, not scared, or ruthless; his actions, verbal aggression, threatened physical aggression, or use of physical aggression; and his likely reaction to the respondent's counter-aggression by backing off, escalating, or trying to kill. Is created a sliding scale of dangerousness, running from low to high. Participants rated how angry and fearful they would feel, as well as how they would respond behaviorally. As the attacker's aggression became more severe, the respondent's decision as to how to respond depended more and more upon the emotional intensity they were feeling. As we would expect, feelings of anger were particularly important in guiding decisions in low-danger situations, while fear was the more influential emotion when danger was high. THE's study's conclusion is that anger functions as aggression facilitator whereas fear functions as aggression inhibitor. Winstock, 2007, p. 131. The tragedy of the World Trade Center provided an unexpected opportunity to examine the effects of anger and fear in a more naturalistic context. In the weeks after the 9/11 attack, a nationally representative sample of Americans participated in a survey about risk perception. Lerner, Gonzalez, Small, and Fishhoff, 2003. The were also asked how angry and how fearful they felt after the terrorist attack. Fear increased risk estimates and plans for precautionary measures, while anger had the opposite effect, decreasing risk assessments and increasing support for an aggressive policy of deporting foreigners. Women reported significantly higher levels of fear and gave higher risk estimates than men. Between 60 and 80% of the sex difference in risk assessment was explained by these differences in the strength of fear and anger. There is also evidence that fear explains the magnitude of sex differences in experimental studies of aggression too. Eagly and Stephan, 1986, asked 290. High stakes and low risks, women and aggression. Judges to rate descriptions of 63 experimental studies in response to the question, how much danger you would face if you enacted this aggressive behavior. Female judges rated the danger of using aggression significantly higher than male judges, and the actual sex differences found in the original aggression experiments were stronger when the sex difference between the male and female judges was larger. In fact, the judges' estimated danger ratings were the strongest predictor of the sex difference in aggression actually found in the studies. TH's was confirmed in another meta-analysis by Betancourt and Miller, 1996, who found the same sex difference in danger judgments, and once again, the size of the sex difference in aggression was correlated with the extent to which female judges perceive greater danger than males. As they note, in experimental studies the ethical safeguards mean that the actual danger is very small indeed. Yet faced with the same low level of objective danger, women found the situation more threatening than men, and this predicted their lower level of aggression. Some research even suggests that fear has stronger effects on women than men. Edelyn Verona and her associates Verona and Curtin, 2006, Verona, Sade, and Curtin, 2009 conducted a series of laboratory studies of aggression. In one such study, Verona and Kilmer, 2007, strong or weak air blasts were directed at the throat of participants as they attempted to complete a reaction time task. Participants reports Confi-RMED that these air blasts caused fear. Following this, they were allowed to deliver electric shocks to a second participant whenever they made errors on a recall task. No surprise that men gave higher shocks than women, but when the results were examined within each sex, a pattern emerged. Women in the high air blast condition gave lower intensity shocks than their counterparts in the low air blast condition. Among men, air blast intensity was not related to shock delivery. Fear reduces aggression in women. Significantly more than it does in men. Emotion in the brain. How does the brain cope with information about provocation and danger from the world outside? Unfortunately, most neuropsychological studies of emotion focus on the experience of just one emotion at a time. But in the real world, we often experience different emotions simultaneously. With jealousy can come sadness. Bittersweet feelings can make us feel happiness and sorrow at the same time. And of course, I have suggested that we often feel anger and fear together. How can the balance between the two emotions be evaluated and weighted so that they are capable of informing our behavior? I have stressed that much of the everyday decision-making that drives our behavior has a strong emotional emotion in the brain. 91. Input. Neuroscientists are beginning to uncover the brain regions that may be involved. And we are beginning to see where the sex differences may lie. Ledoux's pioneering work made it clear that the amygdala was involved in the registration of fear. But since then, things have come a long way. We now know that it is also involved in the processing of a wide range of emotions, both positive and negative. Sergei Chakal and Armani, 2008. Its role seems to be to rapidly detect stimuli that are biologically relevant. Sander, Grafman, and Zala, 2003, especially where they may require an immediate response, which is often the case when they are unexpected, threatening, or dangerous. Adolphs and Spezio, 2006. The amygdala sends this alerting information to a region of the frontal cortex, the ventromedial prefrontal cortex (VMPFC), figure 3.1, and its partner, the ventral anterior cingulate cortex. I should say that I will include studies of the orbitofrontal cortex (OFC) here also. Is may dismay purists, but there is some controversy as to terminology in this region, with some researchers distinguish in the OFC and VMPFC, and others including OFC as part of the broader area. HYP, ACG, PFC, VMPFC, AMI, brain stem. Figure 3.1, emotion and emotional regulation in the brain. ACG equals anterior cingulate gyrus, AMI equals amygdala, HYP equals hypothalamus, PFC equals prefrontal cortex, VMPFC equals ventromedial prefrontal cortex, lying toward the central underside of the brain. The dorsolateral prefrontal cortex DLPFC is not shown separately but is part of the prefrontal cortex. It lies toward the exterior and side of the skull. Reproduced from Toast, H., and Meyer-Lindenberg, A. 2010. I Fear for You, A Role for Serotonin in Moral Behavior. Proceedings of the National Academy of Sciences of the United States of America, 10740-170712, Figure 1, copyright the 27th of September 2010. Proceedings of the National Academy of Sciences, with permission. 92. High Stakes and Low Risks, Women and Aggression. Of VMPFC. There is still a fair amount of debate as to the exact functions of these areas, but they are certainly implicated in the experience and control of emotion, and in our seemingly automatic or prepared responses to these emotions. VMPFC acquires and stores knowledge about the reward and punishment probabilities associated with the emotional information that it receives from the amygdala and sensory areas. Grabenhorst and Rolls, 2011. It can therefore predict the value of approach when the stimulus is rewarding, or avoidance if it is likely to be punishing. Sometimes this information is straightforward and the amygdala requires little additional input from the VMPFC. When it is more complicated, the VMPFC intervenes. For example, psychologists have long been aware of a phenomenon called the framing FECT. Imagine I award you some free money, $50, and then ask if you want to gamble with it. I make you a proposal. you can either keep $20 of it or you can gamble on keeping it all, the gain frame. Sometimes I make a different proposal. You can either lose $30 of it or you can gamble on keeping it all, the lose frame. Of course, from a purely rational perspective, there is no difference. Yet people show a fairly consistent pattern. They prefer the sure thing in the gain frame but are willing to gamble in the lose frame. THs represents the most common decision pattern, and when participants make this kind of choice they show heightened activity in the amygdala. THEY seem to be responding to fairly basic signals about approaching and avoiding risk. But when they occasionally make the much less natural decision to gamble in the gain condition, they show activation in the anterior cingulate cortex, an area allied with the VMPFC, as if this structure is working to correct the natural inclination they felt from their amygdala. DeMartino, Kumaran, Seymour, and Dolan, 2006. VMPFC is able to talk back to the amygdala, turning the volume of emotions up and down. For example, the VMPFC is activated when people actively try to alter their mood. Cooney, Jorman, Atlas, Eugene, and Gottlieb, 2007, or suppress it. Levesque et al., 2003. But as well as regulating the intensity of emotion, the VMPFC can intervene to prevent the automatic behavioral inclinations that are generated by strong emotions. Beer, Knight, and Desposito, 2006. Oxner et al., 2004. Callous unemotional syndrome is a subtype of childhood conduct disorder involving acts that are frequently aggressive. THSA aggressive children show structural and functional problems in the VMPFC. Specify Cali they show a weaker connection between the amygdala and the VMPFC, suggesting that an inability to suppress emotion and inhibit automatic responses may lie at the heart of their problems. Rubia, 2011. Normal adolescents with decreased OFC volumes have a stronger tendency to argue back at their parents during disputes. Whittle et al., 2008. And in adults, an underactive VMPFC and OFC is associated with impulsive aggression. Best, Williams, and Coquero. Emotion in the Brain. 93. 2002, Blair, 2004, Coquero, Sripada, Yanowich, and Luan, 2011. THE involvement of the OFC was demonstrated in an innovative neuroimaging study by Pietrini, Guazelli, Basso, Jaffe, and